maybe let me introduce our first speaker. Our first speaker is Dr. Markus Wöhr, actually professor for biological psychology and behavioral pharmacology at Catholic University Leuven. He is the head of the Social and Affective Neuroscience Research Group at the Faculty of Psychology and Educational Science at Leuven. At present, he is also holding a Young Investigator Group Leader position at the Department of Behavioral Neuroscience, Faculty of Psychology, Philipps University of Marburg, Germany. He has a broad background in animal behavior and translational research models for neuropsychiatric dysfunctions, with specific training and expertise in behavioral neuroscience of affective and neurodevelopmental disorders. His main research interests include neurobiological mechanisms underlying social behavior, acoustic communication through ultrasound vocalizations, and socio-affective information processing in rodents. So he will be carrying on a lecture titled Ultrasonic Vocalizations, Modeling Mania in Rats.
Okay, thank you very much for the invitation and the kind introduction. So first question, do you see the slides? And can you see my mouse?
Yes, yes, we can.
Yes, we do.
Yes,
okay, computer mouse, not, this is a red, okay, good. Okay, so thank you very much. In my presentation today, I will talk about red ultrasonic vocalizations and how they can be used in translational model systems for neuropsychiatric dysfunctions, such as bipolar disorder. Because I'm the first speaker, and actually it's quite an honor, to be honest, I will start with a more general introduction first, and then move on and present what evidence is out there in support of the idea that ultrasonic vocalizations can help us model mania in rats. So there's continued need for animals to advance brain sciences. While there are alternatives to animal research in neuroscience, such alternatives, as probably all scientific methods and strategies, suffer from limitations. So most importantly, in the context of neuropsychiatric dysfunctions, we obviously need a behaving model system, a behaving organism, which gives us the opportunity to experimentally manipulate neurobiological factors underlying behavior. So these alternatives here complement rather than replace animal research in neuroscience. Mice and rats have been the leading model organisms in biomedical research for well over a century. The most recent numbers shown here by the European Union show that about 75% of all experimental animals are actually rodents. This is also true for animal models for neuropsychiatric dysfunctions, and such sometimes controversial standard assays are repeatedly applied. However, modeling human neuropsychiatric dysfunctions in rodents is extremely difficult. And this is because of the subjective nature of many symptoms, the lack of relevant biomarkers and objective diagnostic tools, as well as the early state of what we actually know about neurobiology and genetics. So what's important to emphasize is that the diagnostic criteria for neuropsychiatric dysfunctions are purely behaviorally defined. This means that the validity of relevant rodent model systems strongly relies on their behavioral phenotype. Behavioral phenotyping is the key component here. However, this strategy depends on sensitive behavioral assays. Sensitive behavioral assays with high relevance to each diagnostic symptom category. And this is why I believe that a deeper understanding of the natural behavioral repertoire of rodents and its relevance for modeling human neuropsychiatric dysfunctions is so important. So I'm convinced that this is needed to overcome current limitations of translational research, and that we need to listen to our animal models. So species differ quite substantially in terms of their auditory abilities. We humans, for instance, we are able to hear sound within a frequency range of about 16 to 20,000 hertz. However, there are other species that are able to perceive sound that we humans are not able to hear, because sound frequency is too low for our human hearing system, as for example, elephants, or too high for our human hearing system, as for example, dolphins and bats. dolphins and bats are able to perceive sound in the so-called ultrasonic range. There's actually quite a substantial number of species that are able to hear in the ultrasonic range, and this includes mice as well as rats. So the introduction. Mice and rats are highly social animals. Their rich social behavior repertoire includes the emission of calls in the ultrasonic range above the human hearing threshold of 20,000 Hz, so-called ultrasonic vocalizations. And here you see the three main types of ultrasonic vocalizations emitted by rats, which can be differentiated based on a number of acoustic features, such as peak frequency, call duration, and frequency modulation. For instance, juvenile and adult rats emit so-called 22 kHz calls in aversive situations, such as confrontation with a predator, a cat, for example, or aggressive encounters with conspecifics. It's widely believed that such 22 kHz calls reflect a negative affective state, anxiety, fear, and we, as many others, have actually provided evidence in support of that idea by means of a fear conditioning paradigm. So during fear conditioning, rats learn to associate tone presentations with footshock applications, and as you can see on the left here, freezing behavior, the most commonly used measure of fear in rodents, is highly positively correlated with the emission of aversive 22 kHz calls. However, rats do not only emit 22 kHz calls during conditioning, but actually also during testing. As you can see here, during testing, no footshock is given. Rats are just exposed to the tone they learn to associate with footshock applications, in our case, the previous day. Again, there's a positive correlation between freezing and the emission of the 22 kHz calls, and the fact that rats emit 22 kHz calls in absence of footshock application indicates that this is an emotional response and not a pain response, because actually there's no painful stimulus here. No footshock is given during testing. In support of the idea that 22 kHz calls reflect a negative affective state, we further showed that 22 kHz call emission increases with the aversiveness of the situation, here experimentally varied through footshock intensities. As you can see here, highest call numbers are emitted in response to moderate and high shock intensities. We further showed that interindividual differences in trait anxiety play a role. We screened here in this example a normal unselected group of wild-type rats for anxiety-related behavior on the elevated plasmase, and then tested them in the fear conditioning paradigm. And as you can see on the right, highly anxious animals emit a lot more aversive 22 kHz calls during fear conditioning as compared to low anxious animals. Of course, we were wondering what is actually causing such interindividual differences and decided to study the consequences of early life stress. So we exposed juvenile rats to brief episodes of stress, forced swimming, elevated platform exposure, and immobilization stress, only once and only a few minutes, and then tested them in adulthood in the fear conditioning. And as you see here on the right, animals exposed to juvenile stress, they vocalized a lot more, again, in the fear conditioning paradigm as compared to non-stress animals. We next asked whether this is also true for stressors applied even earlier during life and exposed mothers during pregnancy to poly-IC, mimicking a viral infection. We then found that the animals, when tested in the fear conditioning paradigm in adulthood, that the animals that got exposed to the poly-IC, that these animals again vocalized a lot more in the aversive range compared to saline-treated control animals. This is quite interesting because we did not see other clear behavioral effects despite a very detailed behavioral analysis, including freezing, rearing, and grooming. And this indicates that measuring 22 kHz calls can help to reveal group differences and treatment effects, in this case, poly-IC treatment, that we would not be able to see without measuring 22 kHz calls. In appetitive situations, in contrast, such as, for instance, rough and tumble play or mating, juvenile and adult rats emit so-called 50 kHz calls. And it was Jörg Panksepp, together with Jeffrey Burgdorf, who came up with the quite provocative idea that such 50 kHz calls do not only reflect a positive affective state, but actually share some similarities with human laughter. So while this is a bit provocative, there's actually evidence supporting the idea that these 50 kHz calls reflect a positive affective state, and I want to give you 3 examples for that. First example, rough and tumble play. So I show you a short video, and I hope you will be able to hear the vocalizations, which I downsample, of course. So these are two juvenile rats playing with each other, and while playing, they emit positive 50 kilohertz calls. Similar to the 22 kilohertz calls, 50 kilohertz call emission is characterized by prominent interindividual differences. So here we use the selective breeding approach and selectively bred rats for high and low anxiety-related behavior on elevated plasmase. So an approach quite similar to the one that I just presented to you, but here in this case, it's a selective breeding approach. So we had three different groups of animals. So LAB, low anxiety-related behavior, HIB, high anxiety-related behavior, and NAB is a random control line. And as you probably would expect for a call type that is supposed to reflect a positive affective state, there was a negative relationship between anxiety and 50 kilohertz call emission during rough and tumble play. So they played less, high anxious animals played less, and basically did not emit 50 kilohertz calls, positive 50 kilohertz calls during rough and tumble play. And this is also clearly evident here in this time course graph. So very few 50 kilohertz calls were emitted by the high anxious animals, whereas the other two groups vocalized a lot. So the ones that were not as anxious as the high anxious animals. Second example, tickling. So when mimicking rough and tumble play through tickling, rats also emit positive 50 kilohertz calls. And I really like it. As you see, 50 kilohertz call emission is associated with very strong approach behavior. Third example, and I will focus on this one in the context of today's presentation, drugs of abuse. So rats emit high levels of 50 kilohertz calls when treated with drugs of abuse, particularly psychostimulants such as amphetamine. Call emission is low under baseline conditions, as you see here. Baseline conditions, very few 50 kilohertz calls. The same is true under saline conditions. But then when treated with amphetamine, you see, particularly with moderate doses, 0.5, 1, 2.5 milligram per kilogram amphetamine, we see a strong increase in 50 kilohertz call emission compared to the saline control group. So we actually see several hundred, several thousand ultrasonic vocalizations in response to moderate doses of amphetamine. This is the 45 minutes time course. In response to a pretty high dose of amphetamine, however, call emission is not as high as compared to the other dosages. So here, in response to 5 milligram per kilogram, they vocalize more compared to the saline group, but clearly not as many calls as in response to the moderate doses, because this very high dose towards the end of testing led to stereotypies. And during these stereotypies, the animals did not produce 50 kilohertz calls. Amphetamine-induced 50 kilohertz calling can be dose-dependently inhibited by dopamine D2 receptor antagonists, such as here an example, etigloprid. Besides dopamine, the serotonergic system plays an important modulatory role, and we showed that amphetamine-induced 50 kilohertz calling can be inhibited by co-administering the serotonin 2C receptor agonist, CP809-101, shown here. With the two highest doses of CP809-101 actually completely blocking 50 kilohertz call emission. So they basically did not vocalize at all anymore, despite the fact that they received amphetamine. Next, we applied the serotonin 2C receptor antagonist, SB24-2084, and co-administration of SB24-2084 potentiated amphetamine-induced 50 kilohertz calling, indicating bidirectional modulation of 50 kilohertz calling through the serotonin 2C receptor. This is shown here. So an increase in amphetamine-induced 50 kilohertz calling. To assure receptor specificity, we then co-administered both the serotonin 2C receptor antagonist and the serotonin 2C receptor agonist in addition to amphetamine. So they received three different drugs and found that the two effects cancel each other out, shown here. Targeting the serotonin 2C receptor did not only affect call numbers, but also the prevalence of 50 kHz subtypes. 50 kHz calls can be divided into 4 different subtypes, shown here. I mean, other studies included many more different subtypes, but to keep it kind of simple, we included here these 4 different subtypes. So in response to amphetamine, a large number of calls are frequency modulated calls, step, trill, or mixed calls. And this increase in frequency modulated calls can be reversed by the administration of the serotonin 2C receptor agonist, CP89101, but is potentiated similar to call numbers by administration of the serotonin 2C receptor antagonist, SB24284. We then, when administering both agonist and antagonist, the two effects cancel each other out again, similar to call. This shows that the serotonin 2C receptor is critically involved in regulating amphetamine-induced 50 kHz calling, possibly by modulating dopaminergic activity. Again, as for the other call types, individual differences play a key role, as shown by Stefan Pocinski and colleagues, for instance. So here, one of our own examples. So what we did, we divided rats into rats that emit many or very few 50 kHz calls. So basically we have 2 groups here. LC is low callers, HC high callers. And this is baseline vocalizations, baseline 50 kHz calling. So during conditions without amphetamine. So we used spontaneous 50 kHz calling and divided the animals depending on their spontaneous call emission rate into low callers or high callers. And then we compared them, how do they actually behave during conditions when they actually receive the amphetamine. As you can see here, high callers, they produce a lot more 50 kHz calls than low callers. And this is true for 1 milligram per kilogram amphetamine as well as for 2.5 milligrams. Amphetamine hyperlocomotion, however, did not differ between low callers and high callers, indicating that hyperlocomotion and 50 kHz calling are regulated by at least partially distinct brain circuits. So this has important implications for role models for bipolar disorder. And in this review, I summarized evidence in support of the idea that mania, elevated mood, can be measured through amphetamine-induced 50 kHz calling in rats. So bipolar disorder is a severe mental illness characterized by the recurrent occurrence of biphasic episodes of depression and mania, interspersed with milder but complex fluctuations in mood. In rodent model systems, mania-like phenotypes are typically induced by administering amphetamine. Amphetamine-induced hyperactivity is often used as the only readout for mania-like elevated drive. As shown on the left, locomotor activity here, amphetamine leads to an increase in distance traveled, shown here. And this increase in activity can actually be blocked by co-administering lithium. Can be blocked by lithium, the gold standard for treating bipolar disorder in humans. However, mania is characterized by 2 core features, elevated drive and elevated mood. And I, together with others, suggested that we can actually measure both at the same time by adding ultrasound recordings to our experiments. As shown on the right, amphetamine leads to a strong increase in 50 kHz calling. That's basically what I just told you. And this increase can be blocked by lithium treatment as well, as shown here. This means that we can actually measure both core features of mania, elevated drive, and elevated mood by measuring 50 kHz calls in addition to locomotor activity. So here a summary of the suggested positive relationship between the emission of 50 kHz calls and the intensity of positive affect. While 50 kHz calls are typically absent under conditions with low emotional arousal, substantial rates of 50 kHz call can be observed under conditions with high emotional arousal. This includes rough and tumble play, and depending on the nature of the social interaction, high rates of 50 kilohertz calls might occur, reflecting a high positive affective state. Amphetamine treatment leads to exaggerated emission of 50 kilohertz calls, indicative of mania elevated mood. And this increase in 50 kilohertz calling induced by amphetamine can be blocked by lithium administration, reflecting the dampening of the mania-like elevated mood in line with the mood stabilizing properties of lithium. Supporting evidence was obtained in sleep deprivation studies. It is well known that sleep deprivation can enhance mood, and this is why sleep deprivation is sometimes used to reduce depressive symptoms in humans. So here we showed that sleep deprivation leads to hyperlocomotion on the left side and to a very strong increase in spontaneous 50 kilohertz calling. So this is spontaneous 50 kilohertz calling. This is not stimulated by amphetamine. This is spontaneous 50 kilohertz calling in response to 24 hours of sleep deprivation, and actually a much stronger effect after 72 hours of sleep deprivation. Very high numbers of 50 kilohertz calls, positive 50 kilohertz calls. In support of the idea that exaggerated 50 kilohertz calling reflects mania-like elevated mood, the sleep deprivation induced increase in 50 kilohertz calling can be reversed by lithium treatment in a dose-dependent manner, as shown here. So moderate and high doses of lithium can completely block the sleep-induced increase in 50 kilohertz calling, and actually can also be used to reduce the sleep deprivation-induced increase in locomotion. So here now an overview of studies on the effects of anti-manic drugs on 50 kilohertz calling in rat models of mania. So as you can see, this includes multiple rat models for mania, amphetamine treatment, lisdexamfetamine, treatment, and sleep deprivation. As well as different types of pharmacological treatments. We see here lithium treatment, of course, but also treatment with tamoxifen and myricitrin. What's important is that this pattern is consistent. Anti-manic drugs, at least when given in sufficiently high doses, block mania like exaggerated 50 kilohertz calling, irrespective of how this increase was actually induced. Okay, so in the last couple of slides, I now want to focus on the communicative function of ultrasonic vocalizations. I guess we all agree that rats do not produce ultrasonic vocalizations to tell us humans how they feel, right? So the question is, why do rats actually emit ultrasonic vocalizations? What is their function? And here you see the setup that we established to conduct playback experiments for testing the communicative functions of ultrasonic vocalizations. It's basically an elevated 8-arm maze, which the rat is allowed to freely explore, equipped with 2 ultrasonic speakers. Here's one speaker, here's another speaker. We used ultrasonic speakers to present natural ultrasonic vocalizations, and I guess the most straightforward comparison here is between positive 50 kilohertz calls and negative 22 kilohertz calls. We then analyzed the behavior of the rat, for instance, by quantifying distance traveled as a measure of exploratory behavior before, during, and after playback. So this is the time before playback. This is the 1 minute of presentation, gray area, and then this is the minutes after playback. And as you can see in the graph here, so the playback of aversive 22 kilohertz calls leads to an inhibition of exploratory behavior. So exploratory behavior is reduced not only during, but actually also after playback. As shown in a more recent study together with Markus Wöhr, behavioral inhibition in response to playback of aversive 22 kilohertz calls occurs irrespective of whether the 22 kilohertz calls were induced by predator urine exposure. So in our case, we use lion urine, which is actually not that easy to collect, or were recorded during testing following fear conditioning. And this indicates, so here, behavioral inhibition, here behavioral inhibition, and the summaries. So this indicates that 22 kilohertz ultrasonic vocalizations serve as alarm calls to warn conspecifics about danger. What's important to emphasize is that this is a specific response that is not seen when rats are exposed to phase scrambled, here, and frequency shifted 22 kilohertz calls. This means that there's a relevant frequency band, and only if rats are exposed to calls within the appropriate frequency range, they display behavioral inhibition. The idea that 22 kilohertz calls serve an alarm function is in line with previous results that we obtained in a social fear conditioning paradigm. So what we did is we exposed one rat to fear conditioning, standard fear conditioning, as shown before, and there was another rat in the other chamber, in an adjacent chamber. And the other rat, the observer, was not exposed to fear conditioning, never received a foot shock. But still, what we found is that 22 kilohertz call emission by the sender induced freezing by the other animal, in the observer. And there was a positive relationship between 22 kilohertz call emission in the sender and freezing shown by the observer. Even though, so the observer showed freezing even though he never was exposed to a foot shock. So while the averse of 22 kilohertz calls induce behavioral inhibition in line with the alarming function, 50 kilohertz calls induce high levels of exploratory behavior during playback of 50 kilohertz calls, as you can see here, but also in the minutes after here. So what does the rat actually do in response to playback of 50 kilohertz calls? To illustrate that, I want to show you another short video. So here in the lower right corner is the ultrasonic speaker. And as you will see, actually you see it already now, and the rat shows an immediate orientation response when exposed to playback of 50 kilohertz calls, followed by approach behavior towards the speaker, suggesting an important pro-social communicative function as social contact calls. The way we quantify social approach behavior is through measuring the time the rat spends in proximity to the active ultrasonic speaker. So basically, as shown here in this upper right graph here, we divide the 8-arm maze into 3 parts, into a proximal part, the black part here, the black arms, the arms proximal to the active ultrasonic speaker, the distal part here, the white part in proximity to the inactive ultrasonic speaker, but distal to the active ultrasonic speaker, and then here a neutral zone. When quantifying the time the rat spent in proximity to the active ultrasonic speaker during playback of positive pro-social 50 kilohertz calls, we found that they spent around 40 seconds of about 60 seconds in the 3 proximal arms, and only a few seconds away from the active speaker in these distal arms here. So this reflects very high levels of social approach behavior in line with an important communicative function as social contact calls. We then asked whether amphetamine affects the pro-social communicative function of 50 kilohertz calls and assessed those response effects of amphetamine on pro-social ultrasonic communication in the receiver. So under saline conditions, you see the expected increase in exploratory behavior during playback of 50 kilohertz calls. This increase is only seen during playback, but not thereafter. The increase in exploratory behavior was directed towards the active ultrasonic speaker. So more proximal arm entries, black, than distal arm entries, white. During playback, but not before or after playback. This was paralleled by an increase in the time spent on proximal arms, black, here during playback, compared to distal arms during playback, but not before or after. Okay, so when testing how this is affected by amphetamine, we found that this pattern was actually more prominent in response to moderate doses of amphetamine. So as you can see here, 0.5 milligram per kilogram, 1 milligram per kilogram, 2.5 milligram per kilogram, exploratory behavior is increased and social approach behavior is increased. A high dose of amphetamine, 5 milligram per kilogram, however, had the opposite effect. Exploratory behavior was reduced, and there was no evidence whatsoever for social approach behavior. So we found that amphetamine had an inverted U-shaped effect on social exploratory behavior, but also on social approach behavior induced by playback of appetitive pro-social 50 kHz calls. Social approach was enhanced by moderate amphetamine doses, but completely abolished following the highest dose. Our approach possibly provides a new means to study mania-like aberrant social interaction and communication in animal models for bipolar disorder. So similar to the approach before, we then divided the rats into low callers and high callers, depending on their baseline 50 kHz calls, as before, and compared them across the different amphetamine dosage conditions. High callers appear to attribute more incentive salience here to signals of rewarding social context, the 50 kHz calls, as evidenced by enhanced social approach behavior towards 50 kHz ultrasonic vocalizations, the playback. A response pattern that was also seen in the low callers after receiving amphetamine treatment. So by administering amphetamine to low callers, we can actually turn them into high callers and make them to show significantly higher levels of social approach behavior. And this was true for all 3 moderate doses of amphetamine. So social approach behavior in response to playback of positive 50 kHz calls leads to an increased level of neuronal activity in nucleus accumbens. And the nucleus accumbens is closely associated with reward processing, and a key neurotransmitter in this context is dopamine. Dopamine release is enhanced by amphetamine treatment. And this is why we combine our playback paradigm and fast-scaling cyclic voltammetry to measure dopamine release in nucleus accumbens in response to playback of 50 kHz calls, but also other acoustic stimuli as controls. And to make a long story short, as you can see here, playback of positive 50 kHz calls leads to a clear increase in phasic dopamine release in the nucleus accumbens within a few seconds. No such response is seen during playback of reverse 22 kHz calls on the right. Actually, this is a specific response, dopamine release, only when exposed to positive 50 kHz calls. And this response is actually shown here, positively correlated with social approach behavior. So the conclusions are, rodents emit distinct types of ultrasonic vocalizations. Call emission depends on the animal's affective state, 22 negative affect, 50 positive affect. Call emission is important for communication, 22 serve as social alarm calls, 50 serve as social contact calls. And what I want to suggest is that exaggerated emission of 50 kHz calls can serve as a reliable and valid measure for assessing mania-like elevated mood in rats, and this with sufficient translational power for gaining a better understanding of relevant pathophysiological mechanisms and the identification of new therapeutic targets. I'm convinced that the improved capacity to study the effects of anti-manic pharmacological interventions on a broader range of behaviors, by including emission of 50 kHz calls, in addition to locomotor activity, that this broader range of behaviors will refine rodent models for mania. So I hope I was able to show you that adding ultrasonic vocalizations to behavioral experiments in rodents can provide important additional information. Ultrasonic vocalizations can tell us a lot about our animals, and because of their translational power, we basically include ultrasonic vocalizations in all our ongoing research projects. And with that, I wish to thank my previous supervisors and mentors, Jackie Corley, Francesca D'Amato, Thomas Süthoff, and Rainer Schwarting, as well as key collaborators in the context of today's talk. In particular, I want to highlight the important contributions made by Roberto Andreattini from Paraná in Brazil and Ingo Willon from the Netherlands Institute for Neuroscience. Of course, I also wish to thank the postdocs and PhD students working with me and who contributed a lot of the data that I showed you today, as well as the universities which provided the necessary infrastructure for the studies I conducted in the past and in the future. And this actually brings me to my very last point. There's a PhD student position available at KU Leuven. If you're interested in social behavior and ultrasonic vocalizations and you want to help me filling my lab there with live, please contact me. Thank you very much for your attention.
Thank you very much for this wonderful opening. Okay, we don't have much time for questions. However, there is one on a question and answer, so maybe you'll try briefly answer and then perhaps elaborate on expert discussion panel.
Yes.
So what would be your current thoughts on the reasons for such large inter-individual variability as compared to freezing, let's say? As so many rats don't produce any 22 kilohertz during or after conditioning. So this is a question by Emma.
Okay, yeah, thank you. Very good question. So in my view, two things are happening. The first that happens when the animal is exposed to a mildly aversive situation is that they start to freeze. And then when the situation gets more aversive, then in addition to freezing, they show vocalizations. So the aversiveness of the situation is very important. So it's basically a two-step function. So that I think, so the aversiveness is one factor. The other factor is that there's probably animals that are more likely to warn conspecifics about danger. And what we typically see, for instance, is that call production, the 22 kilohertz call production is much higher in males than females. So it might well be that there's a kind of, they kind of share the work, let's say. Some animals have specific tasks, for instance, to warn animals about conspecifics. And and these animals actually might be more likely to emit these 22 kilohertz calls than compared to other animals in the social colony.
Thank you.
Okay, thank you.
Can I have one question? Are those low callers are less social? Those 50 kilohertz low callers are less social? Mm-hmm.
Yes, I mean, we did not test that in this particular experiment, but evidence from selective breeding studies done by Jeffrey Burgdorf, Stefan Podzinski, and others suggest that this is actually the case. So that when selectively breeding animals for 50 kilohertz calls, that this affects their social behavior as well. Yes. So there's a link between baseline 50 kilohertz calling, so the spontaneous emission of 50 kilohertz calling, and social behavior. Although we did not test that in this particular study.
Thank you very much. Thank you.
Yeah, thank you.